Right. So, in the second hour this week, I'm going to start talking about BLAST algorithm. In the overall layout of the algorithm, the ideas behind the BLAST algorithm. But one of the important things BLAST provides us is not also the algorithm is really nice and it's really it solves a very important problem to have these database searches almost instantly for uh, thousands of sequences. Another thing it provides us in 1990s the statistical significance measure that it computes, the e-value that helped the biologists, researchers a lot on whether the database hit that they have found is meaningful. It may indicate homology or not. So this is very important, and we're going to discuss this actually next this week, today, and the remaining of this week. We're going to talk about this. Those raw scores, how can, as I said, those raw scores do not mean anything if you're using a different scoring matrix or the length of the sequences. And how can we, when we obtain an alignment, for example, that is like this on the slides we have, look at this alignment. Is this really, does this alignment result really mean there is some homology? This part of the sequences are conserved, and does this have some biological significance that we can use to potentially indicate homology between these two sequences? Is what the main question that the main reason we are doing this alignment. Okay, and when you do something like this, for example, if you look at these scores here, the score of 31, 32, 115, which one is good? What does it mean to have a score of 52 among these sequences? There's a local sequence alignment with a score of 52. Does that mean anything? What if I just put here in this list of sequences another row, which is a completely random sequence? If I obtain, for example, score of 52 with that random sequence as well, what does this mean? This means that I could have obtained this one randomly too. So I cannot say this may be biological evidence. So this is what we will do actually. And one of the things that BLAST algorithm, the initial BLAST algorithm, did not have gapped alignments. It was doing, there were mismatches and matches, but it produced local alignments without gaps. And the main reason was not algorithmic actually. When they are doing the extension of this exact match, they could have extended the alignment by introducing gaps. But one of the main reasons they didn't have gaps is that it was difficult. They didn't the statistical significance measure that they were computing. It was be computable only for non-gapped alignments, and so just I'm stating this just to show you that how important that computation of statistical significance is. I mean, because they couldn't compute the statistical significance for alignments that contain gaps, the mathematical closed form formulation was very difficult to obtain if you introduce gaps into the alignment. They just didn't put it into the algorithm because statistical significance computation was difficult for gapped alignments. The initial version of BLAST, if I'm not mistaken, it was just local alignments without gaps, match and mismatches, and they were able to compute statistical significance for the resulting alignment based on the ideas from how the scoring matrices are built in the first place. Okay, but basically the question we will be asking, we are looking for biological significance. Is this alignment, is this match of 26 or 76, this row score, does it indicate a biologically significance? Does it indicate that can there be potentially a homology between these two sequences is what we try to ask. But since we cannot know this biological significance for sure, we will be using statistical significance indirectly to infer biological significance. And the statistical significance will be asking this question: how likely, for example, is to obtain the same score between two random sequences? If it's very likely, the significance means, if it's something is statistically significant, it means that it's very highly unlikely to observe this by chance. Okay, so this is what it means. But if we see that if somebody says you can obtain the same score of 26 between two random sequences, then it means that the match between the cytochrome C human and the hemoglobin beta human is really some random match. Okay, it may be like one of that random match, because if it's possible to obtain the same score randomly. So this is what statistical significance is. And BLAST actually also contributed, the algorithm BLAST contributed a lot in terms of statistical significance to the area of sequence searching. There is another database search algorithm named FASTA, but these days the term FASTA is not used to indicate, to refer to the algorithm. So when people talk about FASTA sequences, it's basically saying a sequence format, a text file format to represent biological sequences. So you may see FASTA, FASTQ, with quality scores, especially with next generation sequencing. We have these new sequence formats. FASTA is more synonymous with the sequence representation format than the algorithm itself, but it's also can be used as a heuristic to speed up database searches. I'm going to talk about BLAST only, and I'm going to just go over it briefly. The main algorithm, I will not go into the details. The basic algorithm, I will just give an overview of that, and then I'm going to demonstrate how a BLAST run by going on to the website, and we will just look at the results and try to interpret what they mean. And after that, I'm going to start talking about how we compute that statistical significance, especially the e-value that the BLAST provides. All right, so it was initially developed in 1990 by Altschull et al., and then different versions of it. It's constantly being improved. I think CyBLAST is 1997, so which dynamically changed the scoring based on the initial results. So there are different versions of it with more biological specificities increased, and different improvements have been done. So there is TX BLASTX, which gets, for example, a nucleotide sequence, then converts it to a protein translates it into a protein and does the search in the protein sequence space. There is BLAST-P, which does the protein sequence similarity. BLAST-N, or it is the default BLAST for nucleotide BLAST to compare nucleotide sequences. It's called a heuristic method for local alignment. Based on some assumptions and idea, it doesn't guarantee optimality, and it's designed specifically for database searches. It's really fast, and it's based on the idea, if we have a good alignment, it should contain some short lengths that we may have gaps or mismatches, but gaps or mismatches should not appear everywhere between every two pairs of between every two amino acids, for example. There should be short lengths of exact matches in a good alignment. So it's based on this premise. And the idea, for example, for protein sequences, I think this short lengths of exact matches is actually very short for protein sequences. It's just three amino acids by default, and 11 nucleotides for nucleotide sequences, because when we have 4 letters, it's a higher chance of random matches. So a random match of 3 nucleotides is quite that probability is a lot larger than random chance matching of 3 amino acids when we have 20 letters. All right, so for nucleotides, this short length exact matches is 11 nucleotides, and for proteins is 3. So initially, This is like the indexing part of our query sequence. Actually, imagine we have already assumed that we already indexed the database. This is our query sequence. First, the query is process, as it's an optional step, filter low complexity regions. Means that, for example, if there's a poly A tail, I mean, this will not be, this is MNS, not a messenger RNA. So, but if there are, I don't think we will have low complexity regions in proteins, but this is usually mostly used for DNA sequence matching. Low complexity regions means that there are certain regions of the DNA with lots of repetitions, like lots of G's, lots of C's. They may not indicate conserved important regions, so they are first taken out, which may be, this step is also optional. But here is the important part. If the database is indexed for 3 amino acid length exact matches, we do the same thing on our protein by sliding a window of size 3, overlapping windows of size 3 all the way to the end. And these are our exact words. We probably compute a hash for that, and this hash, numerical hash, could immediately be matched against database sequences. Okay, so in O1 time, you would immediately know whether which database sequences will contain PGL in their sequences and where. So we are going to find all PGLs in all database sequences very fast. After that, so this is the exact this exact matching parts in the database sequence are called seed. These seeds are then extended for an ungapped alignment. So this is the description of the original blast. So we extend them left and right to see whether this match goes further to left and right, or if even if we have mismatches, we continue these mismatches. Maybe after that mismatch, we will still have a match and the alignment score is going to increase. So we continue this extension until the score drops below a certain predefined threshold. So these are called high scoring words, high scoring pair. So after we do the extension to left and right using ungapped alignments. So this is the original alignment blast where we have ungapped alignments. And the main reason of doing ungapped, not maybe instead of being more algorithmic limitation, it's actually more statistical significance computational limitation. So the extension continues as long as the score increases or stays the same, or maybe drops only a little. This may be a parameter that they may set. At the end, we call this a high scoring pair. Then if we may have multiple high scoring pairs between a pairs of in sequences, and within a distance, high scoring pairs within a distance a of each other are combined together, are joined, and the longer sequence is also then extended as before, and the final result is shown to the user. So the main idea that should stay with you after today's lecture is that so BLAST starts with exact matches and extends them left and right to find the alignment. And since it starts with this local region, it provides a local sequence alignment at the end. And the high scoring pairs, only the top certain top number of them are kept, or they use a cutoff score S to keep only the extended high scoring pairs that have a score of at least S. And the step 6 is really important. A statistical significance score of each high scoring pair is computed. BLAST computes what we call E value for this, and then it reports these to the user. In the original BLAST, we had only ungapped alignments, sometimes joined together to produce larger alignments. And BLAST 2 actually extend the high scoring pairs using gapped alignments. So they develop methods to compute statistical significance for gapped alignments as well. So it's one of the few algorithms to make it as a verb, like Googling, blasting, to run a blast search against a sequence database. Extension is the most time-consuming step, but BLAST 2 was also reported 3 times faster than the original version at the same quality. So I'm going to now go ahead and do an example BLAST run on BLAST website with some sequence. And also, uh, I'm not going to talk about FASTA. Again, it's a heuristic. Again, it starts with exact, trying to find exact matches, finding their um, frame locations, and try to do dynamic programming on this reduced version of the table where we have these long exact matches. But yeah, you're not responsible from the FASTA algorithm. Now, yeah, this one concludes week 2 slides. I'm now going to do an example blast run. We can, for example, try a random blast. All right, we can write our sequence here, long. So again, I have some small letters here. So I shouldn't be that fast. So this is just a random sequence that I'm entering here. You can write their gene accession number here to search for a specific uh, gene that is in one of those nucleotide databases, or you can write your own sequence. As you can see, you can also, or FASTA sequences. Okay, FASTA, as you can see here, is not referred to as an algorithm, but as a sequence format. And it's actually not much different than this one that I'm writing here on the screen. Let me zoom in. Okay, so here is my nucleotide sequence. Okay, these are just random sequences. So we shouldn't get any hits from with a random sequence. Or when we get a hit, the statistical significance should not be more should not be big, okay, I mean it shouldn't be significant. So what is a FASTA sequence? So it's actually the help tells me more about this, but you can learn about FASTA formats by online by looking at resources online. So I'm just going to use the default parameters. So nucleotide correction, the non-redundant database of nucleotides. Uh, there are different databases that you can choose from. For example, you can search against the PDB nucleotide database, which means that these are the nucleotide sequences of the uh, genes whose protein products have known three-dimensional structures. So Protein Data Bank is the database for protein structures. So if you want to search whether this sequence has some homology to a protein structure or to a protein with no structure, you can search against that database. You can search in particular organisms. Okay, so I'm going to skip those parts and optimize for highly similar, somewhat similar, most discontiguous megablasts. And this is, I think, the original BLAST N. So there are different versions of BLAST you can select. Okay, I chose the BLAST N version, and okay, and you just say BLAST. Okay, so let's see what we're going to get. So we are given some job ID, request ID, and it's currently running, and the page is updated like in every now and then, every 2 seconds. Maybe if it takes long, it will be updated less frequently. So it's not as fast as Google search, but we'll see. We'll probably get results in a minute or so. And after we get the results, I'm going to show you how we interpret those results. So 30 seconds have elapsed. As you can see, yeah, the update threshold, the frequency increases as the query takes longer time. Let's see, let's wait. I believe we are going to get some results in a minute or so. It wasn't a very long nucleotide sequence. You can find sequences on NCBI GeneBank or UCSC Genome Browser, or if you're interested in protein sequences, you can go to UniProt database to get those sequences. For example, let's try to find some protein sequences from UniProt. Maybe we can also do a search in that. So there's Swissprot, which contain manually annotated and reviewed proteins sequences. There's Tremble, automatically annotated but not reviewed. There are 200 million protein sequences there. So, for example, if we start from Tremble here, let's try to browse it. Yeah, there are different entries here. These are the organisms we have here. Yeah, the organism and the sequence. Just select on one of them. What is this transposase? If you click on this, there's lots of function information about it. What does it do? What does this protein do? This is an unreviewed protein. It's a predicted protein in this organism. It may have a function in DNA binding. So these are all predicted by using programs, by just looking at the sequence, by finding similarities with other DNA binding proteins, for example. Okay, and here's the FASTA sequence. Okay, actually, this is the sequence which with those numbers are not actually in the FASTA format, but it tells us it's as you can see, arranged in 10 amino acid by 10 amino acid, 50 amino acids in a row, and this is the entire protein. So when you click on this download FASTA, this is the FASTA format. So let me just copy this to my Notepad++. All right, so the FASTA sequence looks like this. The amino acids are not wrapped around, so there are multiple lines of amino acids. I think are there 80 amino acids in one line or 70? I'm not sure. So, but there's probably a fixed number of amino acids on every line. So you just do word count on this to count number of letters. And in addition to, so this is just a text format, just the amino acids are listed here. And there is one header. This is the header of that FASTA format. The header is initiated with this greater than sign. It indicates the header for this FASTA sequence. It tells probably this is the name of the organism. This is its UniProt protein ID and other. So this is the organism. I think species, yeah, different things. And this is actually, you can write free text here, whatever name you want to give my protein. Okay, so after this greater than sign, you write your protein ID. This is understood as a header file that you write some information about the protein you provide. Okay, that's a very simple format, is this FASTA format. Now, what about our results? Yeah, we got the result, but yeah, was it in 1 minute, 2 minutes? So let's see. Query ID, description, none, reports. Yeah, no significant similarity found. For reasons why, click here. We actually know the reasons why. It was a random sequence, right? So we didn't get any results. So let's do a protein blast on our, let's do a protein blast here with our predicted protein sequence. I'm just going to paste here my protein and non-redundant protein sequences. Non-redundant means that if there may be certain proteins with more than 95% sequence similarity, if we have many of those proteins in the database, why we don't need actually all of them to occupy redundant space in the database. So this non-redundant protein sequences select one representative from those highly similar protein sequences, which reduce your database size and hence increase the search time. So we are doing BLAST P. There is PsyBLAST, PhyBLAST, Delta BLAST, as you can see, Quick BLAST P. There are many versions of BLAST, so it's constantly being developed. So now we are doing a protein search. Since this is a real protein with some predicted function, I assume you're going to get some hits, for example, maybe to other DNA binding proteins. So let's see what will happen. By the way, while this is running, I'm also going to start talking about the statistical significance and give you some idea about what we will be doing. So yeah, statistical significance of alignments. Let me download these slides on my desktop. All right. So I will skip this fast algorithm. So the reason I already mentioned why we want to compute the statistical significance, because the purpose of sequence alignment is to infer homology. To when we get some alignment result, whether this is from two random sequences, the aligner, whether it's BLAST or another tool, it's going to try to align any given sequences, whether they're related or not. But it's like we just give two sequences to see whether they're related or not, and the program is trying to align them whether they're really biologically related or not, and it produces some result. Then the next step is just to analyze this result, whether this is something meaningful or not. And the way to do that is to compare it against with what would happen if we just gave it random sequences. So how different it is from random is what statistical significance is about. And sequence alignment is really used for lots of things like function identification. And let's see our blast result. It's here, so it's still going on in 2 minutes. There's been 2 minutes, but it's still trying to do the alignment. So how much similarity is, for example, 10% similar? Another thing is, in addition to the similarity row score you have, you may also compute other measures like percent identity. People use this a lot. These two sequences are 10% similar or 15% similar. So how much similar do the sequences have to be so that we can say they are homologs? Usually 40% of, if they're similar more than 40-50%, we can infer homology, but sometimes the sequences are only 5% similar in very small regions, but they can still be homologs, especially in these divergent sequences. You will not be able to use the row score or the percent identity to indicate homology. Yeah, when the similarity is detected by your algorithm, so when your algorithm reports some alignment result and there are some matches, there are two cases that the similarity is obtained. The similarity can be obtained by chance, or they really are homologs. They have evolved from a common ancestors and hence similar functions. So our goal is to try to distinguish between these two things. Is it by chance or is it this one? The more different, if we can find a measure to indicate that it's very unlikely by chance, then it will probably this other option. So we will never be certain they're homologs, by the way. And when we are computing statistical significance, it's all statistics, and there are probabilistic conclusions that we are going to arrive. We will never be 100% sure that they're homologs, but we will have a strong evidence, very significant result, statistically very significant result. It means that it's a very strong evidence that they are homologs. Okay, let me take a look at this one again. Yeah, we got, I think, some hits. At least the last query ended. This is my query. Yeah, we have some significant results. Yeah, actually, it's the same thing, right? I mean, this transposase is the same protein that we had. It's 100%. This is the max score, total score. So this is our exact match. This was our accession number in UniProt. This is our original protein, and it has also similarities to other proteins as well. Here, the important, which is marked in bold here, is this e-value. E-value is the statistical significance measure BLAST computes, and e-value is not a probability as we go lower. Actually, it's similar to many proteins with e-value 0. 0 means that it's highly statistically significant. So the number of database proteins. So let me describe what e-value is. E-value in the case of BLAST, it means that it's the number of database entries, database sequences, which can obtain same or better score by chance. Okay, it's not a probability, as I said, as I defined it, it's the number of database sequences. It could be, for example, 3. It could be 4. As an e-value, we can see something like 4, 3, but they will be deemed not statistically significant, because it means that the number of database sequences that can obtain same or better row score by chance. That's how e-value is defined. If by chance three database sequences can obtain same better or better score by chance, it means that this may be one of those 3 random by chance matching database sequences. So there's no biological significance here. And as I said, it's not a probability. So you should treat it like, but 0 means that it's really low. I mean, it's the lowest it can get. E-value is something between 0 and infinity. But usually an e-value of 0.001, 10 to the minus 3, is defined as a significance threshold. More than that, for example, even if you have 0.5 as an e-value, which means that only 0.5 database sequences, this fractional number doesn't make sense when you talk about numbers of database sequences that can obtain same or better score by chance. It might not make sense, but it means it's not 0.5 probability. It's like just 0.5 half sequences could obtain this by chance. In other words, if you maybe doubled your database size by 2, you would probably obtain 1 by chance. Okay, so we're counting it, but in this database, we're also counting in fractional numbers. This e-value, you can consider it like this. It's like if you throw a coin once, what is the expected number of heads? It's 0.5 times. It's something like, or something else. If you toss the coin 3 times, okay, now this is going to make more sense than show its difference from probability. If you toss the coin 3 times, what is the expected number of heads you're going to obtain? And you're going to say 1.5, right? But 1.5 is not a number. You either get 1 or 2, but the expected number of heads when you obtain a fair coin 3 times is 1.5. That expected number, that's where this e comes from. That's the expected value. Is, as you can see, a floating point number, and can be some between 0 and infinity, any number. And as I said, the statistical significance, it's a universal measure, independent of protein length. Maybe it changes but for this database at least, it allows us to compare different alignment results. If they are of different length, we would be able to compare the e-values to find whether they are significant or not. But these results actually I cannot show you because there are lots of sequences which are from the same family. They're really similar to this protein. There's 100% identity, right? So this protein is similar to, I don't know how this is the non-redundant database. It seems, I mean, if it has 100% similarities, this is query coverage actually. If you look at the max score, total score, the percent identity is not exact the same. But still, this is 97% identity, 96. It means that if it's 96% similar to many of these other proteins, there are the non-redundancy level is not cut at 95% similarity, probably at 99% similarity. Okay, our non-redundant database that we're searching against, the redundancy may be cut off at 99%. Okay, but anyway, you can try this blast for different sequences, and the e-value is the statistical significance score, and it tells us whether the number of database sequences that can obtain the same or better score by chance. So the smaller the e-value, the larger the significance. And now I'm going to talk about, and we are going to continue on Thursday, how we compute that e-value. And I'm going to show you some a closed form mathematical formula to compute the e-value for non-gapped alignments, just like the original BLAST did. And I will just also then give some ideas how we can be this can be extended to gapped alignments. But yeah, this is the plan for this week. And in order to compute this, whether it's by chance or not, we need to revisit the we need to revisit the scoring matrices, because the value is there, because we will be asking this: if I obtain a score of, for example, 50, can I obtain this by chance? But what does this 50 mean? Why is the match score 5? What does that 5 indicate? We will need to take into account the values in those scoring matrices to be able to identify whether two sequences or the result matches significant or not. We will start with something simple. Before we go to scoring matrices, we will just start this analysis with a number of matches. For example, if I give you two protein sequences of length 100 each, and if I tell you that my algorithm find an exact match of length 5 between these two protein sequences, would you be surprised? Okay, so this is a simpler problem that we will start with this, and then we are going to extend that idea of the length of a matching run to contain the scoring matrix results, and instead of just the number of the size of the exact match, we are going to say I found a match between these two with a score of 50. Is it significant or not? We'll ask that. So let me ask the question here again. So I have two protein sequences A. Both of them are 100 amino acids long. Okay, another B. Okay, so BLAST uses 3 amino acids as the exact match result, right? So if here between these two protein sequences, if I tell you that there's a region here of 5 amino acids, 5AA, and there's a region here also 5AA, they are exactly the same. Okay, now how surprised would you be with this, or how likely this is? Can this happen between two random sequences? All right, so this is what we're going to actually. I can see some of you probably are get yourself some pen and paper and trying to work this out, whether this is something expected or not. I could have asked the question the other way, saying that between these two sequences, what is the expected length of a matching substring? What is the expected length? Expected length of a random match between these two things. Expected length of some matching. So if 5 is, for example, if the expected length is 2, and if we get a match of 5, oh, then it's longer than 2. Probably this is something unexpected. We can say that. But we can now the question that I'm now asking is, what if I have these two protein sequences? What is the length of an expected length of a matching run? Let's call this matching run. Matching run is a substring in both sequences which are exact. So what if they were 1,000 amino acids long? What would happen in that case? What the result probably would change, right? I mean, you would expect more the length of the matching run to be more. Now let's try to analyze this problem. For example, if I select one amino acid from here, what is the probability that another amino acid here will be of the same type? For example, this is of type X. What is the probability that this will also be X? Okay, so I selected this one. These are protein sequences, okay? And I'm going to try to look at if I want to start a matching run at this location, what is the probability that the first letters are matching? Okay, this one matches with this. Is it 1 over 20 or is it 1 over 400? What is the probability that this one, I mean, I selected something here and this one is the same as this amino acid. Okay, or I mean, this may not be fixed, or you may just select the exactly. So 1 over number of amino acids. So Ogün, your answer is correct. The number of amino acids is 20. The number of types of the amino acids is 20. So if you select the random position here and another random position here, what is the probability that they are of the same type? Okay, I'm not asking what is the probability that they are both tryptophans or they are both a certain type of amino acids. Okay, so the probability that they are both tryptophans will be 1 over 400. I mean, assuming that all amino acids are equally likely appearing here, which is not the case in nature. We will come to scoring matrices at that point. But assume all the amino acids, now forget about scoring matrices and assume all the amino acids, all the letters here are equally likely, unlike nature. So the probability of observing a particular amino acid at a particular position is 1 over 20. All right. So the probability that these two amino acids at another position is, if the probability that they are the same, is 1 over 20. Okay, because if you select one of them, it happens to be E, for example. Okay, the probability that the other is also E is 1 over 20. This 1 over 400 is the probability that two amino acids at some particular location are of some fixed type of amino acid. If I ask you, what is the probability that both of them are E's? If I ask you this, you would probably tell me 1 over 400. But over all 20 amino acids, the probability that they're both W's are 1 over 20, 1 over 400. The probability that they're both cysteines, the probability that they're both leucines. So these are all 1 over 400, 1 over 400, 1 over 400, and there are 20 of those. So you add up these probabilities and you get yourself 1 over 20. All right. Now, what is the probability that the amino acids right next to them are also going to be matching? So what is the probability that I'm going to continue this matching to 3 to 5? So for example, probability that to obtain 5 matching amino acids one after another is going to be this, right? 1 over 20 times 1 over 20 times 1 over 20. In other words, it's going to be 1 over 20 to the power 5, right? If you're looking for that. So this is the probability of obtaining 5 matching amino acids at some fixed position. Now, here is the question. How am I going to incorporate the length of the amino acids here? So this probability is like the probability of observing. The probability of observing, yeah, so 1 over 400 is the correct answer to my previous question. What is the probability we will get 2 matching amino acids, then 3 matching amino acids, another 1 over 20 multiplied by 1 over 20. Now, the problem that I'm trying to ask is the question that, is this the probability to obtain a matching of 5 amino acids between these two sequences? No, right? I mean, this is the probability to get 5 matching amino acids in some random starting position. Starting position I here, starting position J here. Okay, the probability that these are matching amino acids is 1 over 20 to the 5. So what is the probability that I would, or actually this is not going to be probability at this point when I start multiplying. What is the expected number of length 5 matches between these two things, between these two amino acids with 1000. Now I increase both of them to 1000 amino acids. So it's like, now this problem is like this. The chance of winning the lottery is maybe like 1 in a million, right? If you buy 2 million tickets, you are like, I mean, what is the expected number of winning tickets you're going to have? It's going to be 2 million times 1 in a million. It's going to be 2, right? You want to expect 2 winning tickets. So if to the probability to have a winning ticket is 1 in a million, and if in that particular lottery, if there are maybe 10 million tickets sold in total and 10 prizes are going to be held out, okay, 10 million tickets are printed, 10 high prizes are going to be given out to whoever buy those. So the chance is 1 in a million. So if somebody buys 2 million tickets, can you say the probability that he will win is 2? No, right? The probability that he will win. So I missed what Ogin. So 0 is the probability to win the lottery. That 0 probability may be referred to that. Yeah. So it's 1 in a million. It may be that. But if yeah, I mean, if it's not a rigged lottery, all right. So if it's really a fair lottery, not rigged, 10 prizes are going to be held out, 10 million random tickets are given out to, well, by the way, we're 5 minutes out of time, so I'm going to stop after we discuss this. But what I want to say is that if somebody buys 2 million tickets with one ticket 1 in a million chance, this will not mean the probability of winning a prize is certain. It's not certainty. There's still, you need to compute the probability. It may be a high probability, but the expected number of prizes that person is going to win is 2. All right. This doesn't mean he's going to win a prize for sure. There's still probability of winning no prize. 0 prize is still non-zero. But we expect that person, if that person says, oh, I won 2 prizes, I'm a very lucky person, we will not treat them as a lucky person. Of course, you won 2 prizes because you won 2 million tickets. You bought 2 million tickets. So with 2 million tickets, we expect you to win 2 lottery tickets. So that's the expected number. If he won 4, that's a little bit unexpected. If he won 0, that's also unexpected, but there are possibilities. But 2 will be the expected number. Now, here I'm going to finish today's lecture with, if I have 2 amino acid sequences of length 1000 each, the expected number of exact matches of size 5, length 5, between 2 of them, just like that lottery example, we are going to multiply it with all the possible ways we can try this match. We can start, when we start this match with a specific I and j, the probability is this. But we can start different i's. Exactly, it's 1 over 20 to the 5 times the, not exactly the length of sequences, but it's like this. We can start I from here, j can be here, then here, then here, then here. Now I can move I 1 to the right, and I can again try all j's, right? It's like the length of the sequences doubled. Like it's like if the length of this sequence is m, the length of this sequence is n. Actually, the exact number of places that I can start trying this match is m minus 5 plus 1. And in the other case, n minus 5 plus 1. So this is, I can start at the first position, and the last position I can start this match is this n minus 5 plus 1 position. It is, I mean, which is 9,996. All right, then 97. There are 5 entries, 5 sequences here. So the last position I can start and try this match of length 5 is this 9,000. Not 9,000. This is 996. 996 position. So, but roughly, when these numbers are large, we can ignore this part actually, which will, I mean, m and n are going to dominate this. So it's roughly m squared, if they're both of the same length, is m squared times the probability of that a match of length 5. So this is the expected value we have. You can compute this for this one, and 1 over 20 to the power of 5 multiplied with 1 million. So it's like we are going to roughly try this exact match 1 million times. It's like we bought ourselves 1 million lottery tickets. In those 1 million lottery tickets, and this is the chance of winning one lottery, how many lottery tickets you expect to win? If that number is 2, 3, 4, then we shouldn't be surprised to find a match of 5 amino acids, right? So that's the same idea. So I'm going to finish today's lecture now, and I will be happy to